This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Today, we're joined by Abbie Smith of ERV to talk about HIV vaccines.
So here's the deal. We've got a multitude of hurdles with HIV that we don't have with other viruses.
And we'll discuss the Immunity Project, a crowd-funded effort to make an HIV vaccine that all of us find a little problematic.
There's all these people who are making the promises of, if you just give me the money, I will give you your HIV vaccine. And it's like, no! If it were that simple, it would have happened 25 years ago. It's not if I just have the money.
We'll also discuss issues with funding scientific research and a whole lot more, so stick around. Hi, this is Audiommunity Episode 9. I'm Kevin Bonham, and joining me, as always, is Matt Woodruff.
Hey,
hi, Matt.
How's it going, Kevin?
What are you drinking today, Matt?
It's the same. I'm sorry I'm being boring today. It's Dewar's. It's
Okay.
just in
Well,
this.
that's
So
fine.
so my
Consistency is good.
Yeah, my trick with Scotch is that if it's not that good, one of my good friends gave me this sort of fancy bottle to put Scotch in, and
So
so
it looks-
you dump the crappy Scotch into the fancy bottle, and then you feel like being fancy.
Very nice.
So,
I like
yep,
it.
That's what I'm doing tonight.
Also joining me with the laugh is Kate Franz.
Hey, everybody.
Hello, Kate.
Hey, Kevin, how you doing?
I'm doing well. What are you drinking, Kate?
I am drinking raspberry Hefeweizen UFO.
Classy. Very
Because
classy. Though tasty.
It was all that was in my refrigerator, and this is not mine. It's Jordan's, because I don't drink these beers.
Fair enough. Also joining us is a special guest, Abbie Smith, who is a postdoctoral fellow at Emory University studying HIV, and she's probably best known for her blog at Science Blogs called ERV, where she writes about all kinds of stuff, including endogenous retroviruses, HIV, vaccines. She tends to bust up creationists and HIV deniers, vaccine skeptics, all of that kind of stuff. So, hello, Abbie.
Hi, thank you for having me.
It is a
How's it
pleasure
going,
having you. What are you drinking, Abbie?
I
You're
am
bucking
drinking-
the alcoholic trend.
I am drinking Stark Sunkist 10. It is a delicious low-calorie beverage. It's very orange, but I am drinking it out of a Sweetwater Brewing Company glass. They have a brewery
Excellent.
down here in Atlanta, and it's really fun. So it's
Very
kind
cool. And
of-
actually, we were just talking before we started recording about the fact that Matt is going to be moving down to your neck of the woods very soon. So maybe Matt will be drinking, in a few months, you will be drinking Sweetwater Brewery things.
Every Friday,
feel like it's
Saturday, they
unlikely.
have like a free tasting thing. Like they do like
Nice.
brewery tours, and you get tickets, and you can try like their seasonal beers and stuff. It is absolutely fantastic.
Very
yeah,
cool.
I suspect you'll be able to find me there then.
Yeah.
Excellent. Well, the reason that we have Abbie joining us is because of something that she recently wrote about on her blog and that we were very interested in discussing on Audiommunity, and that is something called the Immunity Project. Not Immunity Project. Immunity Project.
I didn't realize how much of a problem that's going to be until just now.
Yeah.
Yeah.
So the Immunity Project is a crowd-funded attempt at generating an HIV vaccine. And so they recently published their first paper, and, well, maybe we should save the broader description of that until later, but the short answer is that most of us think that it's kind of not good. But before we get into that, we're going to give you some background on the Immunity Project itself, HIV vaccines, and that sort of thing. So, Abbie, why don't you tell us a little bit about the Immunity Project and why you got interested in it and interested in writing about it?
Okay, well, here's the deal. You guys know funding right now in science is not what it was 15, 20 years ago. This is not the go-go 90s where, just a ton of money was pouring into scientific research. Universities were building brand new research buildings left and right to house all of these scientists that they had and all of these grants that the scientists had. So the funding situation has basically gone downhill. So it's harder for labs to get money. And so some labs have investigated alternative means of obtaining funds. And so, sometimes a piece of equipment breaks and, you don't have that budgeted into your grant. Sometimes you need just a little bit of cash to get some preliminary data to write a grant to get money from the government. So I don't completely begrudge individual laboratories for seeking out alternative means of funding. And one of those ways is obviously, crowdfunding. I mean, that's what people do now. If you want to make a potato salad, you go on Kickstarter and you get $50,000 for it.
I want to say real quick, though, so normally the bulk of funding for science, it comes from the government. At least in the U.S., it's almost entirely the NIH. There's also, the National Institutes of Health funds biomedical research. There's also the NSF, National Science Foundation, that funds a lot of more basic science research in biology as well, but also, physics, chemistry, that sort of thing. And there's also a fairly large number of foundations and sort of nonprofit sources of funding. One of the most well-known probably is the Bill and Melinda Gates Foundation. They fund a lot of basic research, but there are all kinds of small foundations that labs also go to. But funding is tight for all of them because every lab is looking for more funding, so the foundations as well as the governmental institutions are basically getting all kinds of requests for funding. So this is a sort of broad problem, and I guess going to the people, as it were, is not a bad next step.
So in principle, I'm not against the idea.
So yeah, I just wanted to throw it out there just for real life impact. This is the sort of thing that gets hit when things like the government sequester happen. I think oftentimes we as scientists talk about how funding's hard and maybe, it wasn't what it once was and it's glory days and that's great. But from a real life perspective, what that actually means is when people do things like sequester government funds, when people cut money to the Department of Health and Human Services, all of these sorts of political things that I think go under the radar unless you're personally affected by them, these are the sorts of things that are actively contributing. So if anyone's out there thinking, well, why is funding going down, all those things, all of the crap that's gone on in the US government over the last 20 years and the complete inability to put together budgets, all of those things, these things are directly impacting the way that science is done in this country.
Absolutely. I actually had a friend who got a job, I guess, at the NIH right before the sequester last year. And so she just graduated with her PhD. She didn't get any funds to help with moving expenses. So she basically spent all of her savings to move, put, a deposit in on her apartment, all that sort of thing. And she gets there and she doesn't get a paycheck because of the sequester and the government shutdown. So she was living off ramen and just crossing our fingers. Seriously, that they were...
I sort of do that now.
Yeah. But no, that the government was going to get its act together and she would be able to collect a paycheck to do the job she was hired to do. So
yeah,
absolutely.
And that's certainly a problem, but we should also say that, even absent something like the sequester, just because doing research is getting more expensive, so there's inflation in the cost of doing science. And so even if even if the amount of overall funding stays the same, if it's static, you actually have a decreasing ability of labs to do science and a decreasing pay lines. I mean, you can look at graphs of the sort of grants that are getting funded, just going down and down and down and down. So it's not just in these extraordinary circumstances. It's also just because we're not increasing funding for science is having the effect of decreasing research. So
okay, well, back to the Kickstarter theme.
Yeah, we're really bad at transitions on this podcast.
Yeah.
We figure it out eventually.
It works.
Usually it takes 2 drinks.
So what you were saying before, the NIH, Bill and Melinda Gates Foundation, all of those organizations dole out the money based on proposals that people send in and preliminary data that people send in. And then they are evaluated by a panel of scientists who say, hey, I think this is a really good idea. This is a good investment of the cash. Or I don't think they put enough thought into this, let them think a little more resubmission, or this isn't a good idea right now. Let's not give them funding. So who gets the cash is evaluated by a panel of scientists who are experts in that particular field.
I'm sorry to interrupt again, but I just want to say that at this point, because of the funding climate that we're in, a lot of times those funding decisions aren't, this is a bad grant, we're not going to fund them.
Yeah.
It's that of
we don't have the cash.
100 really, really great grants filled with amazing ideas, we can only actually fund 20 of them. So let's look for any excuse not to give them money, because
that's a
lot of grants, there are bad grants that are being submitted and aren't getting funding, but there are also a lot of really great grants that are not getting funded because there's not enough money for them.
And I think 20 out of 100 is a really good number. I'd be happier with that number. It's more like 6 out of
Yeah.
100.
Yeah.
Really,
the funding
really
line
good
is about 6.
grant.
Yeah.
Yeah, and it's disproportionately basic science too that gets hit, right? So the difference between basic science and translational science is often that the public has a direct interest in the outcome of the science itself. Unfortunately, scientific discovery doesn't really work like that. Just because you have an interest in something and you fund 14 grants for the same thing doesn't necessarily mean the solution is going to come out of that with higher frequency than if you allowed people to do some really basic science. But that's sort of, I guess that's my own shtick. I don't have
No,
to
that's
go
a-
down that road.
That's a
agree
larger
with you.
conversation.
100% with you though, Matt. Like I think translational biology is having a moment right now, and there's all these people who are making the promises of, if you just give me the money, I will give you your HIV vaccine. And it's like,
Yeah.
no! If it were that simple, it would have happened 25 years ago. It's not if I just have the money. We have so many unanswered questions that the translation
Absolutely.
isn't capable of happening yet. So, yeah, I agree with you 100%,
I'm
Matt.
so with you on that as well.
And that is a great segue into, maybe we can do segues.
Immunity Project.
Yeah, so the,
immunity. Can we just call it hack HIV for 2 reasons?
Oh
First
my
of all, because it
gosh.
sounds less like immunity, but also it sort of typifies how I feel about this group.
Wait, isn't that what they have? Isn't that one of their hashtags or something?
Oh,
Yeah, exactly.
No, they use it, so I feel comfortable in us using it. Abby doesn't seem thrilled about the idea of
No.
even saying hack HIV.
Oh man, that's wonderful.
yeah, maybe they didn't think that hashtag through too much.
Well, and that's what
Does
they are,
anyone
though.
ever... Go ahead.
Well, that's what this group is. This is a group of marketing and business individuals. The people affiliated with, like, if you go to the main the Immunity Project webpage, you are greeted with this video of all these different people saying, "What if? What if? What if? What if we could have an HIV vaccine? An HIV vaccine for free, for free!" Like it's all these different faces saying the same script, and it's like.
That was really good.
Yeah, that was
really, really good.
It may not be apparent to those of you listening, but those were all Abbie voices.
Yep.
I'm quite impressed by
And
that.
there were faces.
There were faces.
You know,
But-
the thing that infuriated me about that what if thing was, if there were an HIV vaccine, I can almost guarantee it would be free. Like,
Yeah.
there's no way, like, the amount of pressure they put on companies to make the drugs available and free or at crazy reduced prices, there's no way you would make an HIV vaccine and then not give it to the people where the greatest burden of HIV was. I just, like, I saw that. I was like, it's gonna happen! Ah!
HIV drugs. It's important to point out HIV drugs.
Right, that's what
Wait,
what
did I
she
not
said.
say drugs?
No, you didn't say HIV. You said pharmaceutical companies are being forced.
But we're talking about HIV.
Oh,
so
yeah. The
Yeah.
pressure.
No, I just wanted to make
yeah.
sure that people weren't thinking that we're defenders of the pharmaceutical, right? That's the...
No, just like the sort of pressure that recently was on Johnson & Johnson to release their top of the line antiretroviral therapy to
Yeah.
like at a reasonable cost so that it could actually go to people where HIV is infecting. And
Sure.
unlike the United States where HIV is kind of under control, we're all good here.
Well, and there's government programs to get people the medication. Insurance covers the medications. Like we have alternative avenues here in the United States that are not available to people in Africa and Southeast Asia and the areas that desperately need the medication.
Right. So, Immunity Project,
Yeah.
Yeah.
So-
good
HIV.
marketers.
Yes, and that's what it is. It's marketing, and it was incredibly light on the science. And I've always worked in HIV vaccine design from an envelope. We're going to have to do some basic HIV science.
Do it.
go.
So there are two main proteins that people designing HIV vaccines are interested in. One is the envelope protein. It is on the outside of the virus, and it is the only target for neutralizing antibodies. So if you want to have a B-cell vaccine, you are going to be targeting the envelope protein. Alternatively, there's an internal structural protein called GAG. It basically makes up like the viral core structure. And it is internal, and it gets processed by the cell's proteasome and presented in MHC class 1 in infected cells. So there's other HIV proteins that are also targeted, but I'm going to simplify it down to GAG. And so in theory, with a CTL-based vaccine, you could induce anti-HIV infected cell responses in that way.
So I just want to point out real quick, we'll have links to a bunch of Abbie's posts that describe the things that she just said in more detail. And also you can check out episode 2 where we talk about antibodies and antibodies against HIV and go into a little bit more detail on that. But
right. And you mentioned the acronym CTL.
Yes.
We've talked about those before, cytotoxic T lymphocytes, right? So these are the CD8 killer T cells, basically. And we have discussed that before, and I'm sure we'll have that information up as well.
Right. So I primarily focus on envelope, but I have looked at it as well from the CTL GAG, or this other protocol, but GAG perspective. And so I was looking over their website and I was like, what the heck are these people proposing? What are they proposing from a scientific standpoint? And
you were looking at it back, was this like a year and a half ago?
No, it wasn't so long.
No, it was like January, February.
Yeah, it was like January. But I was digging around trying to figure out what the heck they were proposing. And so, I looked up the investigators involved. They had absolutely no connection to HIV research whatsoever. Because normally you can look up their previous publications and figure out what's going on. But he had nothing. And so
there's an anesthesiologist from Mass General. I'm concerned about him because he's like right next door.
Really?
Sort of.
I thought, but aren't they all based out of San Francisco?
Yeah, except for the one anesthesiologist out of Mass General.
Isn't he like the head guy?
Yeah. Well, he's the first author on this paper.
Oh, okay. Yeah. I said, yeah.
Why is an MD a first author on a paper?
I mean, maybe he did the experiment.
Interesting question.
Let's not necessarily get into our...
No. We'll have to go...
medical doctor versus philosophical
after
doctor.
Dark. That's Audiomunity After Dark topics that are burned after we discuss them.
We usually turn off the mics.
Okay. But anyway, I this is my field, and I was trying to figure out what the heck they were proposing from their Kickstarter information, and I could kind of guess on what they proposed to do, but I didn't know. So as opposed to, what happens through a normal funding agency where you propose your science, you tell them exactly what you're going to do and how you're going to do it. In this Kickstarter, it was a commercial for a free HIV vaccine with no explanation on what they were going to do. There was no way for a potential funder to say, hey, I think this is a scientifically valid idea, or you're full of crap. There was no way for anyone to scientifically judge this project. And I think that was by design.
And you talked with other people, I imagine, about this who similarly are in your field?
So I believe it was Bruce Walker. Let me double check. There were a number of well-known CTL HIV researchers who were listed on the Immunity Project page. And I was about to email them being like, dude, what the heck is going on here? And before I could even hit send, I went back and checked the page again and all their names were off. So the marketers did not get permission from those researchers to say that they were behind this particular project. And they were the only ones who had any HIV vaccine credibility. When the Kickstarter first came out, they had no publication. And they were going around saying, oh no, first they were saying we need $40 million or something for human trials. And I was like, dude, you don't even have a cell culture paper out. You don't have a mouse paper. You don't have a non-human primate paper. And now you want tens of millions of dollars to do human clinical trials. Like, what the heck was going on? And so in a different interview, he was saying, well, we needed X amount of money to finish these mouse studies. And then it needed X amount of money. Oh, the mouse studies were done. They just needed to get the publication. Like, you got a dozen different stories from this group of individuals. But they finally published their mouse work, and that's the paper.
Yeah, and so that's the paper that was just published recently, and the paper that we're going to be talking about today. And so we should introduce that now before we go on.
So I'd actually like to back up just before we start introducing this paper. And Abbie mentioned, I think, that this Kickstarter idea, or the crowdfunding idea, is an interesting idea. There clearly is a problem with current funding in the sciences. And I fear that with this podcast, we're going to come across as angry scientists lashing out at bad science and lashing out by default at the idea of people becoming interested and the idea of people putting resources behind something that they feel is important. I took a look through, and there have been a couple people that have made this work in reasonable ways. So there's a story out of Duke, for example. Kathleen Pryor out of Duke was looking for funding to sequence the genome of ferns. And she was doing that for, first of all, basic science interests, which I understand. Also, she was looking for potential applications for nitrogen fixing in the soil, all of these sorts of things. We don't have to go into the detail of it, but basically she was having a hard time getting funding through the NSF. It sounds like she turned to a crowdsource model. Some people were able to directly fund the experiments, and as a result of that, she was able to sequence the genome of the fern that she was looking at, right? But there's a difference. And I think the difference comes from exactly what you were talking about. The difference comes from her laying out, first of all, exactly what the project was, exactly how much it was going to cost, exactly how she was going to go about it. And on top of all that, there was a reasonable expectation, because she had built up her reputation within that field, that she would use the money in the way that she was talking about using, right? And so as a result, she crowdsourced it, she got the money, the experiment happened, and now she has data, which has been published and peer-reviewed and all of those things.
Well, and let's also point out that that also sounds like a project that you don't have hundreds of labs around the country competing to sequence that fern. Like,
sure,
there weren't other people working on this, and it's something that she couldn't get funding to do herself. And
right.
it's not that it was conceptually super flawed. It's just that it was something that agencies weren't willing to give money to.
Like you said, funding is tight. The NSF and the NIH have to decide which projects they are going to prioritize over which other projects, right? And it sounds like in this case, people weren't willing to fund this particular experiment. So she found people that were interested in seeing the experiment carried out. They contributed money. And so I guess I just want to make sure that it's clear before we get into this paper that I would absolutely encourage people to get directly and personally invested with scientific causes that they're interested in. I think that the crowdfunding model has promise, although, it hasn't gained a ton of press yet. Hopefully that will change. And I suspect that we're going to have a couple issues with this paper, and I would hope that as we review this, we don't turn people off from getting engaged in this system and potentially opening up future.
It's a very good point. And I have a question, though, on that topic. So the fern thing, that's interesting because that's a tiny interest group, right?
Sure.
But let's think about things that people might be more interested in donating to directly because they have some direct interest. So think about something like multiple sclerosis or ovarian cancer or something like that people want to give money directly to labs.
How about just my research project? Just everyone make it rain on Kate.
So for that sort of thing, where you have a lot of labs that are writing grants to the NIH for, let's just take multiple sclerosis as an example. So somebody wants to study a particular treatment for multiple sclerosis. Does it make sense for that person to go directly to people who aren't experts to, put out a call on social media to anyone that's been affected by multiple sclerosis and try to raise money?
Well, I think that this is what some of these larger organizations really are for. You mentioned the Bill and Melinda Gates Foundation, for example. Bill and Melinda Gates do a fantastic job of bringing in a ton of money, and obviously they raise money amongst wealthy donors. You can give to the Bill and Melinda Gates Foundation as, a normal person. But what they do in order to make sure that that money is doled out correctly is they bring in people very much like the NIH and the NSF. They bring in people that are familiar with these systems, with these fields. They ask for grant applications. So people apply to the Bill and Melinda Gates Foundation.
And this is sort of exactly my point. Yeah.
So they vet. So
right.
So
and
I
this
agree.
is so I think I appreciate the potential novelty of crowdfunding, and I think it's an interesting idea, especially for very small scale sort of, something like that fern project. Yeah, neglected areas of science that probably aren't going to get NSF funding or NIH funding. I think that makes a lot of sense, but I think it makes more sense to have places where people can go. Like it would be cool if there was a structure like Kickstarter, but for crowdfunding of scientific research, where it was like, we're going to have some people vet that the basic science is good before you actually get, like, anybody can start a Kickstarter for anything. Like Abbie mentioned potato salad, like you can do random stuff and it doesn't actually have to be efficacious. I think that if you're going to be funding science and expect any sort of results, you need to have someone that has some scientific expertise actually looking at the project and saying that it makes sense. That's just my opinion.
So I'm looking
and no one's going to refute me. Okay.
Well, so I'm looking at a project called experiment.com currently, where it appears that researchers, I'm going to literally read you the points that I just came across in the 10 seconds you were talking. Researchers post projects needing funding on Experiment. They share their project to fundraise and spread awareness. Once a project reaches its goal, supporters are rewarded with the insight behind their science. So to your point, there doesn't appear to be a peer review process on that. But I think that if you had a system like this where there was a peer review process, and so people could maybe weigh in on the validity of the experiment, perhaps. I don't know. I'm not quite sure how it would look, but it sounds like, or it looks like from here, that people are at least trying to move in that direction.
Just a reminder, though, if you are a U.S. taxpayer, some of your money is going towards this research. I am 100% paid by the NIH. The research I do is by the NIH. They get their funds from taxpayers. So if you want to give above and beyond that, we need to figure out a way, like you said, like a peer review Kickstarter project. I think that sounds fantastic. But you also don't need to be guilted into donating more to a project when you already are.
Yeah, what you should do instead is guilt your Congress people into increasing funding for the NIH and NSF.
Congress people.
And we could get into a long and involved argument about the role of government, but that's not what this podcast is for.
Please let's don't.
So instead, let's
if you do live in a state that maybe doesn't, or you have a congressperson who maybe doesn't support funding the NIH, please call them and tell them to fund the NIH to a reasonable level this year. Or your governor seems to not understand that fruit flies have been a pretty important genetic model.
Okay, Matt, let's not.
No. I have like real stakes in this now. I'm just waiting for a grant to get funding, and I just need the funding to be approved.
It is based on fruit fly immunity? Yeah.
No, it's not. There's nothing to do with fruit flies. So if you're against fruit flies, it's all good. Don't worry about it.
But John Chow would be upset about that.
Oh yeah.
That's true.
Okay. But so
now we should introduce the paper.
So this immunity project, they raised $1.2 million.
That's a lot of money
in crowdfunding.
No joke.
Yeah.
1.2 million.
Yep.
Oh my God, I could buy so many fruit flies.
I have them for free.
And the paper that they published recently in the journal Vaccine is called Eliciting Cytotoxic T lymphocyte Responses from Synthetic Vectors Containing 1 or 2 Epitopes in a C57 BL6 mouse model using peptide-containing biodegradable microspheres and adjuvants. So that's quite a mouthful. The first author is R.M. Rubsamen, and the last author is D.E. Heckerman. And I think we sort of talked about who some of these people are a little bit earlier. But this paper...
This is published in Vaccine, as you mentioned. It's also published as a brief report. And I have not yet looked up what the requirements are for a brief report in Vaccine. I only point it out because oftentimes in journals, there are a couple different ways that you can submit articles to journals. And depending on how you want to submit the article, things like how many figures are in your article, how many words do you want in the article, what do you want the peer review process to look like, they can differ depending on how you submit it. So I don't actually know what the requirements of submitting a brief report to Vaccine are.
Right. So the overall layout of the paper is basically setting up an idea for an HIV vaccine, right? So the idea that they raised money on in this crowdfunding campaign was that they were going to develop an HIV vaccine, and then because it was crowdfunded, they then weren't going to charge anything for the knowledge about how to make that vaccine. And the idea is vaccine disproportionately affects people in sub-Saharan Africa and Southeast Asia, often very poor communities. And so having a vaccine that's free to distribute would be great. As Kate said, even if a pharmaceutical company developed it, that probably would happen anyway. But leaving that aside for a moment, that was sort of the main proposal. And this is supposed to be the first step along in that process. But I think before we get into exactly what they did, I think it would be nice to talk a little bit about, Abbie sort of started talking about this, but approaches to HIV vaccines and why they haven't worked. Because over the last 30 years, we have spent millions and millions and millions of dollars on HIV science, most of which has been to try to get a vaccine. And when HIV was first identified as the cause of AIDS, scientists were really excited because they thought, well, we have the virus now, we'll get a vaccine in no time. And that was back in the 80s. So
2 to 5 years.
Yeah. So
it's just a virus.
It should be fine. So what the hell, virologists? Abbie and Kate. What's going on here?
So here's the deal. We've got a multitude of hurdles with HIV that we don't have with other viruses. And so I mentioned before, envelope, the protein on the outside, the target of antibodies. This protein has a remarkable amount of plasticity. And that means that it can tolerate a lot of mutations while still maintaining its function just fine, which means that if you've got an antibody that neutralizes this virus really well, the virus doesn't necessarily care because it can change that target and replicate just fine while evading that antibody. And the HIV population can do that because everybody knows HIV mutates a lot. And so you don't just have one virus in an individual, you have a population of closely related viruses. Each one of them are slightly different from the other. So you might have an antibody, for instance, that can neutralize, 90% of HIV variants, but then you have 10% of a viral population that replicates just fine. Most proteins are not like this, and indeed most proteins in HIV are not like this. So the targets of antiretroviral medication, the mutations necessary to escape those antiretrovirals normally come at a fitness cost, which means the proteins don't work as well if they can escape that antiretroviral. Or in the case of CTL immunity, the mutations that allow the virus to escape CTL recognition also come at a fitness cost. And so that's why when you give someone antiretrovirals, the viral load drops down. They don't progress from HIV infection to AIDS. But unfortunately, due to the mutational capabilities of HIV, it starts accumulating what are called compensatory mutations, which means it can escape whatever that selective pressure is while still having high fitness. So the deal is with an HIV antibody vaccine is that we just can't make an antibody via vaccination that can recognize the spectrum of HIV variants that someone can be exposed to. So if we...
And we... Sorry, Abbie, to interrupt. If you want more details on this, Abbie has a ton of posts on this, as I mentioned, but we also talked a little bit about that. Was it episode 2? I already shouted that one out.
It was the Michelle Nussenzweig paper looking at the broadly neutralizing antibody cocktail that they were using to drop HIV down to baseline.
And so that was a way of using antibodies, but they were engineered antibodies, and we can't get a vaccine to make people make really great antibodies for
the really great antibodies are freaks.
Right.
They aren't normal.
Yeah, and I feel like we went into that a little bit in that episode. It's too complicated to go into right now.
Oh, I don't think so. I mean, we used... Abbie, you've mentioned antiretroviral therapy a couple times, right? And the reason that you use multiple antiretrovirals is that if the virus mutates around any one of those antiretrovirals, then you have another one that does a different job, and so it just kills the mute, right? And so this is why you use cocktails of things, and I think we discussed this extensively in that episode.
Right, but I'm talking about the fact that those really great antibodies that are really good, even the 7 that they used in that paper,
right?
We can't make a vaccine.
They were natural, but we
can't make a vaccine
that
to
came
force
from each
people
person
to make that antibody.
Right. No, you're not going to be able to elicit that sort of response naturally. Right. You're correct.
Right.
So that's why we haven't gotten an HIV vaccine. So, for instance, if you put virus X into a vaccine, that individual will make great antibodies to virus X, and if they're exposed to virus X, they'll be just fine, but nobody is going to be exposed to virus X. They're exposed to variations of that virus, and they will not get complete protection. So that's the problem with the antibody response. The CTL response is a bit more interesting in that there are mutations that allow HIV to evade the cytotoxic T-cell response. However, GAG it doesn't have the same plasticity as OMS does. And so personally, I have a question as to why the CTL-based vaccines haven't worked. And perhaps they haven't because they haven't induced enough cytotoxic T-cells that recognize the HIV peptide. So along those lines, to draw it into the...
So I'd like to jump in. So this is actually one of the things that I'm really interested in, and I've discussed this before, is the basics of vaccine design from a really basic immunological perspective. And I would argue, certainly you're right in that we haven't haven't done a great job of stimulating CD8 T cells. We've actually done no job at stimulating a solid CD8 T cell response against really anything.
Yeah, really, basically
way.
every single vaccine that we've made that works really well is an antibody-based vaccine.
Right. And even if we did, and this is one of the things that I was going to bring up later when we actually discuss the paper a little bit, but even if we did, even if we were able to produce CD8 T cells in whatever lymph node is downstream of where you injected it. So let's say you vaccinate someone in the arm. You've got CD8 T cells that get stimulated in the axillary lymph node, presumably under the armpit of the person that you vaccinated. Now you have activated CD8 T cells that are going to go back to the site of infection, right? So your effector CD8 T cells are not like B cells. They don't produce antibodies. They can't provide you systemic protection. What they have to do is they have to actually find the infected cells themselves. So even if I vaccinate you in the arm, I'm going to give you HIV protection in your arm, presumably, and not necessarily through the normal route of infection. And this is something I think that's being addressed a little by Akiko at Yale.
Well, and then the problem is as well with CTL responses is that for a CTL response to work, you have to have infection. Well, with HIV, you really need to stop that initial infection because once replication starts going, it gets ahead of your immune system and then you're just playing catch up. So I'm not sure whether CTLs are the best way to go, but that is what they're attempting in this paper.
Yeah. This was my whole problem with HIV vaccines in general, in that like every vaccine that we've designed, it's designed to allow for the initial rounds of replication of a virus or bacteria, and then it can drop the hammer on the infection. But HIV, once it has that initial round of replication, HIV integrates into the genome of a cell. So you have all these cells that are latent reservoirs, carriers of HIV. And so once you stop the replication, you know, like HIV can still continue to replicate. So you need something that can like really stick around and like clear out the entire reservoir, which in itself is a very large problem. Interesting idea, but
Can a CD8 T cell effectively recognize and kill virus-infected T cells, other T cells?
Here is a problem with CTL-based research. Many of the assays that are done are not 100% natural, I would say. So you would think to do this assay, you would take some CD4 positive T cells, you would infect them with HIV, you would mix them with CD8 positive T cells that in theory recognize your peptide of interest. You would assay cell death from chromium release or something like that. Those assays are not done. What they do, and again can pull us back into this paper, is one way they do things is peptide pulsing. So you will take any kind of cell. So all cells have MHC class 1 on the surface, and some of those class 1 molecules will either be empty or you can get peptide exchange. So you mix the cells with the peptide you're interested in, and now all of a sudden you've got lots of MHC class 1 on the surface. Then you can mix
with the peptide that they're specific for.
Then you mix in your T cells of interest and see if there's cell death. Okay, you think that's not entirely artificial. That's a way to measure whether you have T cells that respond, but that doesn't tell you that that actual peptide is expressed on the surface of infected cells. What that tells you is that peptide can be loaded in MHC class 1. And so I was working with a lab in Oklahoma, William Hildebrand's lab, who they do these great big cell farms that they infect with West Nile, or they'll do breast cancer or something like that with secretable MHC class 1. And then they do mass spec to figure out what peptides are actually presented. And when they do HIV, they don't find the rockstar HIV peptides. And then this isn't published yet, but I've subsequently done some experiments that, you know, some of these peptides that the CTL field knows are there, I can't find them. I couldn't find them with my experiment. So there are many problems, I think, with the CTL branch of vaccine design, but what they were doing in this particular paper for the Immunity Project, to pull it back over, is that
I told you the tangents were a problem.
Yeah. No. This is all relevant. So what they were proposing in this paper is basically an elaborate kind of peptide pulsing. And so you wouldn't just inject somebody with a peptide and really expect this peptide pulsing protocol to get you a lot of CTL response. We do it in the lab just for that particular assay I was mentioning. So what they're suggesting is they've got this awesome nanoparticle something or other that if you mix your peptide with that and then you infect infected or injected into someone, then you can get CTL responses. And in fact, they did, not with HIV peptides, but with some standard laboratory peptides. So I mean, they haven't
albumin, immunologist's best friend.
Yeah, so they haven't touched HIV at all. So this paper has nothing to do with HIV. All they showed is that they can do this elaborate peptide pulsing mixture and get a CTL response. What they didn't do was compare it to the standard in the field. Right now, the standard for eliciting a CTL response via vaccine is usually via some kind of viral vector. So you get an adenovirus, you get a retrovirus to deliver the gene of interest, and then that gene is transcribed, processed, everything endogenously in a cell. So instead of giving the cells a peptide, you make the cell produce and process it and present it itself, which would be a more natural situation because, like I said, you can give cells peptides that they don't actually present and get them to present.
That's really interesting. Wait, can I ask a question?
Yeah.
I guess more for myself. So HIV controllers, which is something that the Immunity Project talks about, they say, we're going to use the CTL peptides that controllers present because controllers are thought to have a particular allele of MHC1, which for whatever reason the theory is that they can present these peptides that are really great for CD8 responses. They clear the virus, they can control it. So do we know if HIV controllers actually present the peptides that they're thought to present?
What we do know is that there are regions in hypothesized MHC peptides that change upon transmission of someone with one MHC allele to another MHC allele. And so if you've got someone who is B57, which is a protective allele, they will have mutations that make that virus less fit to escape, hypothetically, the CTL response. When they transmit to someone who's not B57, you see a reversion back to the higher fitness variant because there's no longer that CTL pressure. So I think in some patients, there's absolutely CTL pressure, but you have to have that protective allele. I can't change your genome to make you B57 if you're not. And
until we get CRISPR-Cas online.
But even with but like B57 is only a correlation. Like there are people who are B57 positive aren't controllers. So it's
absolutely
like these weird B alleles and something else, maybe a weird virus. That's kind of like that's still up in the air thing.
Right. Well, and we can't discount, just randomness because the randomness of a quasi-species, the different kind, the permutations it can explore in one person that might stumble across the right mutation early and the next person it doesn't stumble across it at all. So that's part of the problem is there is no genuinely protective allele where you're infected, your CTLs clear the infection, you're cured. That still doesn't happen even with the protective alleles. And then you've got the problem where there are some MHC alleles that actually make an individual more susceptible. They progress more quickly to AIDS after they're infected with HIV.
I hadn't actually considered the importance of what Abbie just said,
No
which
way.
is that I didn't realize just how faulty this idea is, right? Because if you don't have the correct MHC molecules and you are naturally infected with HIV, you're not going to produce the peptides that they're trying to vaccinate with.
Yep.
Yeah, that never even occurred to me. So thank you for that.
Yeah, no, that's...
They explained it so succinctly on their website by just saying magic, superheroes. So you would never think that wasn't a possibility.
But can I go back to the even more fundamental question about CTLs in general? And that is, you say that we think that there probably is CD8 pressure with certain MHC molecules.
Yeah.
But it sounds like that hasn't even been demonstrated, that for sure CD8 T-cells can kill infected T-cells in macrophages that are infected with HIV.
There is evolutionary evidence that there are in some people. So
in some people.
Yeah. So there is no reason why these mutations would occur. These less fit populations would succeed unless there was CTL pressure.
Okay, but that's in these individuals that have this specific MHC haplotype.
Yeah.
And I should explain what I mean by that. So we've mentioned before, MHC is the thing that's actually presenting bits of protein on the cell surface, and there are all kinds of different versions of MHC. It's the gene with the highest diversity of alleles in human population. Pretty sure that's true. Matt, you look skeptical.
I think that's true.
Wait, what did you just say?
Guys.
I said that
it's the most
MHC is the most
diverse.
diverse.
Yeah.
So
I thought you were saying that B was the most prevalent.
No. So do we have an estimate of how many people have this particular allele of MHC?
It is the least common allele of MHC, which is an interesting.
Okay, and Abbie, that's the only allele for which we have evidence that there's CTL pressure?
So there's a group of the B alleles. I don't know if I'm the B haplotypes. So it's like a couple of the 50s, a couple 27s. There's like a couple different alleles in there that are protective, but they're very rare alleles, which I think is interesting in that, so like they're the most rare, so maybe that's just a coincidence, or maybe HIV is just less adapted to these very rare alleles as like a world population virus. You have to do this.
Yeah, I mean that would make sense.
This hand
right?
motion.
So you're gonna evolve to not be responded to by the masses, but every once in a while you're gonna run into a really rare allele, and you're not gonna see them enough to evolve around it. Is that sort of what you're saying?
Yeah.
Okay,
I'll
so
take that.
getting back to the paper, so let's set aside for a moment the fact that no one has demonstrated that this particular approach is likely to succeed or even feasible. Let's set that aside for a moment and just look at what it is that they actually showed in this paper. So we've alluded to it a couple times, but the procedure that they're taking is to load these little microspheres with peptides and adjuvant. And we've mentioned these things a little bit in previous episodes, but just to recap a little bit, most vaccines contain at least two components. One is the thing against which you are vaccinating. So in the case of most antibody-driven vaccines, you have some protein that you want to make antibodies against, usually something on the cell surface of a virus. Sometimes it's toxins of bacteria, things like that. So you have some protein that you want to make an antibody against. And then you also have something that triggers the innate immune system. And we mentioned this in episode 3. We talked about TLRs and pattern recognition receptors. You have something that's going to activate the immune system to recognize that the other thing is dangerous. So normally when we're affected, the first thing the immune system sees is there's something that's infectious, and then it sees something specific, a protein that pathogen contains. So in this particular case, they're doing something similar. They're saying, okay, we're going to have peptides, we're going to have pieces of protein of HIV. Well, actually, as Abbie mentioned, there's actually no HIV in this paper. They're using ovalbumin, but fine, it's a protein. And also
NVSV, a cool virus
NVSV.
that I work on.
Good point.
It's a useful virus.
And then some ligands, some things that are recognized by the innate immune system, TLRs. So they have CpG and MPLA. So that's a TLR9 and TLR4 ligand. And what they show is that if you have a microsphere that contains ovalbumin, you don't get a very good response. But if you have TLR ligands, you get a good response. So that's figure 2. Figure 1 is just like we have microspheres.
Some conditions on that. Well, they have some conditions. First, they try to find out in figure 1 what size the microspheres should be in order to get dendritic cells, which are ultimately going to be important in this situation. So what size the microspheres need to be in order for the dendritic cells to eat them, right? So what they find is that dendritic cells are pretty good at eating microspheres, especially the 11 micron microspheres that they will be using for the rest of the study, right? And that's important actually because later, and they do mention legitimately that people have suggested that large microspheres are not able to stimulate a cytotoxic T lymphocyte response. So it's actually important as to what size microspheres that they are using, although they do argue later in the paper without actually comparing any results that their results directly contradict other people that have also tried to interpret this sort of data, but neither here nor there. So they do at least have a reason, I think, for showing which size they would use.
Right.
But there are also some requirements that they level in figure 2 as far as which microspheres work and which ones don't. And I think it's worth mentioning just because this is, a good thing to think about when you're developing a vaccine. Now, one of the red flags that I had when reading this paper was that they basically say, well, other people have shown that it's important to have TLR agonists inside, but we find that you also need TLR agonists outside when you're doing the injecting. So in this scenario, you would have a microsphere that has the peptide in question. It would also have TLR4, TLR9 agonists inside the microsphere. And also there would be TLR4, TLR9 agonists outside the microsphere.
Right, but can I just say that it doesn't appear as if they controlled particularly well for the dose of TLR agonist that was being delivered.
Agreed.
So when you try to load a microsphere with CPG or MPLA, not everything that you put into the solution is going to get into the microsphere. And they didn't really show that they're delivering the same amount of this ligand.
So I've done a little tiny bit of work. If you remember early on in my PhD, I was working on nanoparticles. And one of the really interesting problems with nanoparticles is that you can always make them. They're pretty easy to make. They're really, really hard to know what's actually inside of them when you make them, which is also a reason why the FDA has a really hard time coming to terms with the idea of a nanoparticle. Because let's say you make these nanoparticles and 10% of them have what you want actually inside of them. The other 90% aren't going to, which is why I almost laughed when they talk about the incorporation of different peptides into these nanoparticles. The idea that the FDA at any point is going to allow you to just swap out peptides at will as if they're the same thing is left, right? The FDA will absolutely
but we don't care about the FDA. We're going to give this away for free to Africa.
Right. No, I know. So the idea that you can just do this and it's going to work and it's going to be accepted and you're just going to inject it into people like this is terrible. But I do want to, before we move on.
But time out. There are FDA approved microsphere drug release technology. Like,
yes,
it's a
and
thing.
You have to
it exists.
Very rigorously prove exactly what's in that microsphere. And if you're going to switch out one of the major components, for example, the thing that you're vaccinating against, you're going to have to reprove every time. And so one of the reasons that they talk about why this will be really streamlined is because they would be able to just switch these things out without re-engineering the microparticle itself. What they fail to mention is every time you switch something out, you're going to have to go back through all of the macaque testing, all of the primary human testing, right? Everything that leads up to the dissemination of that drug.
And it would be different for every single person who got the vaccine because every single person has a different combination of MHC alleles. And so what peptides would need to be included in the vaccine would be different for virtually every person.
And so this sentence really struck me, and this is what I was sort of getting at when I brought this up, and I just wanted to point it out because it really, really bugged me. So I just mentioned the lead-up sentence, which was, in addition, although previously published findings show that increased CTL responses are in the micro, blah, blah, blah. It's important that TLR4 is outside as well as inside the microsphere. The following sentence is, we believe, and they point out that this is contrary to previously published results.
Okay.
But they don't describe why their findings might be different, right? They just say, this is different from what other people have found. The following sentence is, we believe MPLA in the carrier solution acts to stimulate the tissue macrophages in the area where transformation to dendritic cells takes place.
What?
After which phagocytosis and antigen presentation occur. And that doesn't make any sense. None of that. Not a single part of that sentence makes any sense.
I'm pretty sure that I blocked that out when I read it because
I didn't catch that either.
Oh my God.
So that sentence either is really poorly written, in which case they've basically glossed over the entire mechanism of why their findings might differ from the current literature, or it demonstrates a complete non-understanding of how innate immunology works. One of those two things is true.
Well, and I think there is a lot of, they're naive, the individuals involved in this research. So
if you're saying that macrophages are turning into dendritic cells, I think naive is a very, very generous way of putting it.
A lot of this. As to if you think you are going to go with this paper to any funding agency and they are going to give you $40 million for a human trial, I mentioned in my post, you're delusional if you think that's going to occur. Just the simplicity of this paper, it's fine for basic foundational research if you think that this vaccine is the way to go. But they are so far away from an actual vaccine. They're so far away from actually describing what is going on in their proposed protocol.
Well, as Matt mentioned, we were talking about this last week, and the only assay that they're doing to compare their different things, to compare their different conditions, is the production of interferon gamma in PBMC. So that's peripheral blood monocytes.
Is it? Because let me direct your attention to the early results for a second, where, let's see, sorry. So the first paragraph of the results is the only part of the methods in this paper. There are no methods associated with this paper, but they give one sentence to the methods that are used in the entirety of the paper. And in that sentence, they say, fresh splenocytes were harvested and subjected to interferon gamma ELISpot analysis by strict Streeck-Fram-Walker criteria. So I looked up that paper. First of all, there's a problem in that the figures all refer to peripheral blood mononuclear cells, as you mentioned. What we're talking about in this one sentence are splenocytes. I don't know. I assume that they were talking about splenocytes. If they're talking about splenocytes, I A, don't know how they got the cells, and B, don't know why I've got a CD8 interferon gamma producing T-cell population in the spleen when it should have all been in the draining lymph node. Now, if it is the peripheral blood, that's fine, but that means that your only method sentence in the entire paper, the only way for somebody to reproduce this, is 100% wrong.
Yeah, so there's so much to say on that. Okay, so
my problem with it initially is that interferon gamma production is not cell killing. So that was my problem, was that it's a proxy assay, it's not a genuine assay.
Right,
but I didn't even catch that.
Yeah, so let's just recap for a moment. So first of all, the thing that they're measuring, interferon gamma production, is something that, yes, CD8 T cells can make interferon gamma, but so can a lot of other cells. So they're looking at the production of a cytokine that is associated with a particular type of immunity, but actually doesn't tell you anything about the underlying immune response. Like, it tells you that interferon gamma is being produced, but it could be being produced by NK cells, it could be produced by CD8 T cells or CD4 T cells, or like any number of cells can make interferon gamma. It doesn't tell you anything about whether there are actually CD8 T cells that are activated and killing cells containing this peptide. Second of all,
so the method sentence, really quick, just to cap that, the method sentence refers to a paper that talks about the relevance of using interferon gamma ELISpot approach to assess CD8 T cells. They make no mention in the paper of which cells you should be doing this sort of thing on. Basically, what they do is they lay out how you would put together an interferon gamma ELISpot and why that is one reasonable approach to go about this. Yeah. In order for that to be reasonable in this scenario, you would have to, first of all, tell me which cells you're sorting, because, like I said, you don't know if they're from the spleen or the blood, but you would have to put them through a flow cytometer, determine that they are CD8 T cells that you are actually looking at, and then do the interferon gamma ELISpot. Now, I noticed recently that their cytometer just came
good.
On their blog.
How nice.
Their flow cytometer just came in, so maybe it has a sorter on it, and maybe they'll do this in the next line.
So oh God. So going past the fact that they're not actually looking at what they say they're looking at, the second thing is that, as Matt mentioned, they're claiming on the graphs that they're looking at one type of cell, a peripheral blood mononuclear cell. And so I should clarify, when you're looking at human responses, looking at PBMCs is a really great approach because drawing blood from someone is much easier than taking out their spleen.
Or spleen biopsy. That hurts. Spleen biopsies hurt.
So, but all of these things are done in mice, and there is no reason that you would do peripheral blood mononuclear cells, or at least there's no reason you would do only peripheral blood mononuclear cells when you can pull out lymph nodes, you can pull out spleen, you can pull out any number of organs in a mouse and look at what's being done. But as Matt said, the methods say that they are looking at spleen, but they're claiming in the figures that they're looking at PBMCs. So, and that's the only thing they look at is interferon gamma by ELISpot, which isn't even the best way to look at interferon gamma production. It's just all kinds of bad.
So the nice thing about ELISpot though is that at least you're getting information that the interferon gamma production is happening because you've pulsed a peptide, right? So it's unlikely that you're getting an inflammatory monocyte in their study. One thing that you mentioned though earlier is that a CD4 T cell, which would also be responding in an antibody response, could also be producing interferon gamma, right? And could also be a contaminating part of this culture. Yes. And I want to point out, they do mention later that it would be good to do both sorting and cytotoxic T experiments.
Yeah.
And maybe some dose response.
Yeah.
But $40 million for a human trial first would be super cool.
Right, that would be good.
Can I ask for $40 million to inject humans with LPS and see if they generate an immune response?
Could do
see
that.
if there is
Alternatively,
inflammation.
what you could do
Well,
is
and here's
ask
the deal.
Alternatively, what you could do is ask for these people to publish their budget. So they raised $1.2 million for this in an open source funding situation, right? Now, presumably they've been keeping records, and this science is expensive. I don't know how these experiments were done. I know that they just received a new flow cytometer in the lab, all of those things.
So they spend half their money.
So the question really is like this, and Kevin and you and I were talking about this. These experiments in and of themselves they don't tell you much. There's some really basic immunology of vaccines in here, and the one thing that they do show, I think, in the last figure is that they can put two different peptides into their microspheres and get responses to both. Now, I should know in the literature if that's been done before. I know the last paper they cite for that is like 2008 and say that it hasn't been done before. I don't know if it's been done before or not. Regardless, I do know that there are some real big holes in vaccination immunology, so maybe that helps to start to fill one of those gaps if you were to confirm that with like 16 other experiments, right? But beyond that, what you've got here is a summer project.
Yeah, I was going to say, no one's getting their PhD off this paper.
No way. No way. And I'm stunned, absolutely stunned that Vaccine, which is, I think you guys would admit, a reasonable journal. They publish reasonable science. I am stunned that they would accept a paper like this, which is why I was interested before in what the brief definitive report requirements are for Vaccine.
Yeah, I would say not very high.
Apparently. Apparently that's true.
I don't know.
Abby, you were going to say something that... Oh, sorry.
Oh, no, it's fine. Go ahead.
Abby, you had said, here's the deal, and I wanted to know what your deal was.
Oh, I've forgotten the deal.
Oh, I'm sorry.
We've moved on from the deal.
Okay, here's my deal.
I remember the deal.
Steal my deal, thunder, Abby. Steal my deal, thunder. Go ahead.
This is extraordinarily basic research, and I don't say basic in a bad way. I mean, this is basic foundational research. They're not going to get a huge grant off of this as preliminary data, not only because they don't have the history in the field, but this isn't sufficient to get this isn't sufficient for an R01 from the NIH. And rather than saying, you know what, we're new at this, we think we have a good idea, but we don't have enough data yet to go to the traditional funding agencies, what the Immunity Project did is on their webpage, they accused the funding agency of like being in the pocket of big pharma, Big Brother, whatever, won't give them money because this is a radical new idea and they don't want to fund radical new ideas. They really kind of get into conspiracy theory bullshit, to be perfectly honest, as a reason for why they can't get funding in traditional ways. And when you look at this paper, there's no way anyone is going to give them money with something this simple.
No.
Yeah.
No, not at all. Not at all. And
it makes me so mad. It makes me so mad because, as we said, there are so many, like, if the NIH receives 100 grants, as we mentioned before, they can fund 6 of them. And maybe, like, of those 100, like, only, 85 of them are reasonable. Maybe only 30 or 40% are worth funding. Let's say that maybe 30% are worth funding. They can only fund 8 of them. So you have so much good science, really, really good with great preliminary data that is not getting funding. It makes me so angry that you would say, we're just not getting funding because of some conspiracy bullshit. Like, this is bad.
That upsets me
Here's what
too.
they actually said. Furthermore, private equity investors don't like HIV vaccine plays because the real money in HIV is in developing new lifelong therapies that people living with HIV will have to take for the rest of their lives. Big science controls most of the rest of the funding that's available, and our breakthrough IT-based approach to HIV vaccine development is still too early to resonate through the old boys funding network. That is off of their page.
I love the term big science. This is the first time I've ever heard that.
Yeah, right?
Big science.
Most
we are
scientists
so
are a bunch of...
influential.
Big science is actually just made up of communist hippies who brew their own kombucha, but also hold those purse strings tight and keep out those innovative ideas.
IT-based approach.
Oh,
So in all fairness, I would say that there is a bit of a good old boys approach to science. There's no question that people know people. You know, it's been a small industry for a long time, but for anyone that has spent any amount of time reviewing grants, reviewing papers, like basically doing all of the work, right? Because we do all of our own work, right? So we try to publish in these journals, but ultimately the journal calls us up as peer reviewers and says, is this science good? Should we publish it, right? So we are the ones doing the work. We're the ones reviewing the papers. And ultimately, we're also the ones that the NIH will call up and say, is this grant good? So
and that's worthwhile because we are the ethics.
Right?
We are the, sorry to interrupt. We are the experts in this. And that is why the NIH asks scientists who are doing the research to evaluate proposals. And of course, if people are friends, they're more likely to fund their friends' work or people that are doing similar work to themselves. Scientists are human.
This idea that a newcomer couldn't come in from the outside with a great idea and expect that they would get some sort of reasonable review out of the people in the field is already, especially in HIV, the field is already at war with itself over funding. Just because you can't jump into that pit with a good idea and survive the initial onslaught of,
even if you had a good idea, it's not necessarily a good idea.
Thank you for that, Abbie. Right. Yeah, no, even if it was a good idea, but let's stipulate that these people clearly don't have a good idea.
Well, and part of the reason, a way around that is to get into the club, get collaborators who are in. They don't have any collaborators who have any connection with HIV research. If you're a smaller dog, a smaller fish in the pond, you need to associate yourself with a bigger fish. And they decided to go at it on their own. And furthermore, there are means of funding high-risk research. So we've got R21 that are short. You don't ask for a ton of money, but you ask for some, and they're higher-risk ventures. You go to the Bill and Melinda Gates Foundation. They will fund higher-risk novel therapies. That's really kind of what Bill and Melinda Gates want.
Or you go to DARPA. DARPA will fund weird stuff that probably isn't going to work, like only because it sounds weird and it's like high-risk, high-reward.
Yeah.
I love the idea of DARPA.
DARPA scares the shit out of me.
I love the idea
but let's not get
DARPA.
on that tangent. Let's just, I mean...
But
I like DARPA.
But there are places. There are places for risky, interesting ideas. Like at the end of the day, like this isn't interesting. If you look at the field of HIV vaccines or therapies, there's a lot of interesting stuff. Maybe you read it and you're like, okay, this is probably not going to work. But it's different ideas. Like let's take CMV and turn it into an HIV vaccine. Probably going to kill babies, but you know what? Let's see what it does in monkeys.
Let's go for it. Yeah.
But so the conclusion of this, I mean, the conclusion that I reach from reading this paper and from this discussion is that the people that are conducting this research lack basic understanding of HIV, they lack basic understanding of the immune system, and they lack basic understanding of vaccine approaches in general. And
funding, how to go about getting funding.
Yeah, I mean, and
the sense of entitlement here, the sense that, somehow you've stumbled across something that no one else has ever thought of, and despite 30, 40 years now of solid, hard research, you're going to do it better with no resources in your lab, with no personnel that have any experience with HIV. There's a sense of entitlement there that very much bothers me.
The word I used was delusional.
Yeah, no, and I think that's accurate. Did you guys listen to the This American Life episode from a couple weeks ago? I think it was a rerun, but where they talked about the guy who was basically had an idea of physics.
Yes.
And he said that he was basically going to disprove E=mc².
Yes.
And he brought it to physicists.
How'd it go?
And he brought it to physicists, and they were like, yeah, but the equations don't work. And he's like, yeah, to understand my idea, you don't need to understand math. You don't need to understand differential equations. It's like, well, no, actually, to do physics, you kind of have to know those things to be able to say anything about what's going on. And he was convinced that he could show that all of the physicists in the world were wrong about a fundamental tenet of their science. And it feels like that's what this paper is sort of doing, that on a slightly smaller scale than E=mc², to be fair, but they don't understand the basics, and they think that they're going to upend the science, and it just doesn't.
I mean, even how it's written, you can see that they don't really understand exactly what they're talking about. They explain like CpG as like bacterial DNA, and I'm like, okay, someone hasn't read Kevin's paper.
Thank you, Kate.
So there's that.
I also have the feeling from their acknowledgments in the paper that they've reached out to experts in the field and they are not interested in working with them.
Yeah.
No, Dan Baruch specifically expressed both support and interest.
Well, that's because Dan Baruch is actually working on this exact vaccine candidate and probably was like, oh, what are you doing with microspheres? Let me just make sure it's not what I'm doing with microspheres. Oh, it's absolutely insane? Okay,
Yeah.
bye. Continue on your way.
I reached out to people in the Baruch lab, and granted, it was last minute. It was very last minute, but I did not receive a response.
So there's a postdoc who's working on like a very similar idea of like releasing, proteins through these microspheres. Apparently the microsphere, this is the other thing that they don't talk about. Is like one of the interesting things about this microsphere, which I think why the lab is interested in looking at them, is that you get delayed release of your antigen over time. So you can keep this antigen in the bloodstream for a longer amount of time than if you just like shot in someone's arm with like LPS.
Yeah.
But
so go ahead.
So their microspheres actually are antigen, like the polymer is an antigen, is the adjuvant, which is interesting.
Yeah.
But
That is
so
cool.
there's always a conflict of interest disclosure, and I wasn't going to bring this up because I didn't think it was relevant, but now I'm sort of agitated, so I'm going to bring it up. There are 3 people, 3 of the 4 authors on the paper. Now that you've brought up this time release idea that Dan Baruch's lab has been working on with these microspheres, there's a conflict of interest statement stating that 3 of the 4 employees are shareholders of Flow Pharma Inc., and DEH is an employee shareholder of Microsoft. Microsoft thing, not so important. Flow Pharma Inc. is a time release company. They look at the time release of gentamicin. I believe that is their first product, and they're looking to incorporate a product where when you close up surgical sites, you introduce gentamicin time release capsule or something along those lines for what I believe to be legitimate therapeutic benefit, right? So you have basically slow release of antibiotics at a surgical site, which would ultimately help you prevent infection.
Whoa.
But the fact that they're on it and so adamantly arguing against the
Big pharma.
The villainy of big pharma while they're directly investigating studies that are similar to what they're making money on.
Yeah.
Weak sauce.
Yeah, no, it's
weakest of the sauces.
unlike the habanero beer that I had. It is weaker than that.
Hottest of the sauces. And you can get sauced.
You can get sauced on the saucy.
On the hot sauce.
We're just killing time until Kate gets back. Is there anything else that you want to say about this paper or about this project that we haven't gone over? Anyone?
No, I mean,
Bueller?
just what they were requesting from I, as an HIV researcher and as someone who maintains a blog that tries to educate the public about viruses and vaccines and that sort of thing, there are a few things that irritate me more than scientists running out to the general public saying, we cured AIDS, we cured AIDS, we cured AIDS. Because I have to turn around and I get the reputation as a Debbie Downer because I have to write the blog post that said, we didn't cure AIDS, guys, here's what actually happened. And it is absolutely
You do that very
infuriating.
well, though, Abbie.
Well, and I'm annoyed at the rate at which I have to do it, but it's not just these people with the Immunity Project. It's also, universities that want to plug the fantastic research their scientists are doing, so they issue these sometimes outlandish press releases for interesting papers and sometimes not so interesting papers. But to capitalize on the general public's desire to do good and to get an HIV vaccine and to villainize the people who are actually working towards that goal in the process, it just really grossed me out.
Yeah, that's pretty
In
awful.
principle, there's nothing I have against a Kickstarter program to get a lab, a flow cytometer because there's a new laser because one of theirs burnt out, unexpected expenses. I'm not against that. I am 100% against the way this particular group chose to go about getting their funding. And just going back to my original post, some of the outlandish things they said. Now keep in mind the paper that we just read, that we just went over. They said 6 months ago, we are raising $25 million to begin phase 1 FDA human trials. It is now, they're ready. They're ready for a phase 1 human trial in the US and South Africa. As soon as we raise adequate funding, we can begin the study. So once phase 1 is funded and the FDA deems us safe, we will need to raise another $20 million for phase 2 trials in order to test the efficacy in sub-Saharan Africa. Off of this paper, they were telling the general public that they needed $45 million. They were ready to go. They were ready to pull the trigger on this, but those evil scientists were standing in their way.
I'm strongly resisting the urge to throw my mic into my monitor right now. Oh my God.
If you do it, I'll narrate it. It'll be epic.
Both my monitor and my microphone were paid for by Harvard Medical School, so I'm not going to do that.
Yeah, thank you. Thank you, Harvard Medical School.
Okay, well.
So now I feel gross inside.
Yeah.
Yep, deep down in my heart.
You should just drink Andre. Like me.
I have a bottle of it in the fridge downstairs, but no one ever comes to visit me.
What is Andre?
It is a bubbly champagne-like beverage.
It's delicious. Sorry.
All right.
But I would also like to second everything that Abbie said.
That will be the title. I would like to also second everything that Abbie said.
Yeah. And
well, thank you, Abbie, a lot.
Thank you very much
hanging out with us.
for joining us.
It was great. You guys get it. It's a pleasure to have a conversation with people who get it.
We like to think that we get things. And on that Debbie Downer note, this has been Audiomunity Episode 9 from emunity.org. You can find us mostly on iTunes. Eventually, we're going to get on other podcast networks, but that's taking us some time. I'm on Google Plus and Twitter. Abbie is not on any social media, but you can find her at her blog, scienceblogs.com/ERV. And if you're not reading Abbie, you really, really should, if you have any interest at all in being interesting. We have stuff on Facebook that Kate has still not done anything with.
I've done it.
But we have facebook.com/audiomunity exists. Whether there's anything useful there, that's another question. Blame Kate.
I knew it was going to come up today, too, and I was like, fuck, I haven't done anything yet.
But Matt and I are designing a bunch of graphics for this episode, and they also go with previous episodes. We've been way behind on graphics, but you can find those graphics, hopefully some cool animations, and links to Abbie's blog, all kinds of good posts, as well as links out probably to Immunity Project, just because we're going to be nice about it. All of that stuff is at emunity.org. That's EMMUNITY.org. We're supported in part by Harvard Medical School, and the music at the beginning and the end of the podcast is composed by Rachel Reineck. And we'll see you next time.